Hi everyone, and welcome to part C of chapter 8. We're going to extend on the themes that we were talking about last time, where we can see enzyme activity shift when its shape is changed for reasons like of either temperature or pH. But what we're going to do now is talk more about deliberate control by the cell in order to affect whether a reaction occurs or how fast a reaction occurs. Certainly pH can be a controlling factor, but it's less often a controlling factor than a circumstance. Same goes for temperature. But there are some things like post-translational modification and some other circumstances. I'm not really sure what this magnet means in this picture, but we'll roll with it. I was borrowing the picture. But certainly the light bulb analogy here is true because you can do modifications to an enzyme to turn it on and turn it off. So let's get into the control side of enzymatics. We've talked about this a little bit before, but I think this statement says it very, very well, in that you would have chaos if all you did was turn things on with enzymes. You really do need to tightly control, just like you have other controls besides your gas pedal in a car. You have steering wheel, you certainly have brakes, and that's what's going on here. It's not enough just to turn a reaction on. You have to control the rate and whether it's on at all. And there are several overlapping mechanisms to do that. Several overlapping mechanisms. One is whether or not that protein exists in the cell. We have 30,000 some genes. We don't turn them all on at the same time. And later on in the semester, we're actually going to talk about some of the mechanisms that sort of drive this. In any case, you can turn genes on and off using proteins that regulate gene expression. Call those transcription factors. But in any case, it's a slow control. It is adjusting the abundance of a given enzyme or protein. You might remember in the last lecture I talked about if you had one firefighter fighting a fire, they could only throw so many buckets of water. But if you had 200 firefighters, now you've got more, a faster reaction of getting water onto the fire. And that's what abundance is. We sometimes call that expression, if you will, how much a gene is expressed. Sometimes we call this upregulation and downregulation. Again, it's not just up, it's also down. We can slow down a reaction by reducing the amount of enzyme. The other side of this, so we can control from in a faster way, that is modifying the shape or the things that influence the enzyme or the protein that is doing a function. We talked about allosteric inhibitors previously, but there are also allosteric cofactors that will activate. Okay, so we have allosteric regulation. This is outside the active site. That can control how fast that enzyme can go. If you remember that graph with the KMs, some of these things will make the KM go higher. Some things will make the KM go down. Inhibitors will make the KM go down. Cofactors and activators will make it go up. We can also do a more permanent type of modification. If this is potentially transient, and this is much more permanent because we're talking about a covalent bond, that can also, and it says on or off, well, that's not entirely true. We can also modulate how active that enzyme is by adding or subtracting functional groups. So let's look at some of these aspects. So you remember last time we talked about cofactors? A lot of the and inhibitors, well, we're kind of making it more of a gray area here because we have activating allosteric activators. They're cofactors, okay? They enhance an enzyme's activity, and we have allosteric inhibitors. They can bind in different places on the same protein. Call that allosteric regulation. And what's happening is it's adjusting the shape, and that changes the activity of the enzyme. So when a cofactor inhibitor binds a protein at one site and affects the protein function at another site. And the complexity can go way up right here. We're going to try to keep this as simple as possible. There are multiple sites of regulation on many proteins. This is why I was squirming a little bit in the last lecture, because we were talking about cofactors and inhibitors. Well, we're talking about cofactors and inhibitors, and now they're sort of contradicting. The book is contradicting itself. Cofactors will cause gain of function. Inhibitors will cause loss of function. If it's allosteric, it just means it's somewhere other than the active site. It's just that simple. We don't need to kind of do this either or thing. Enzymes do cycle between active and inactive forms depending on how they're modified and what state they're in. Sometimes we activate and sometimes we inhibit. What they're saying is when you have an activator, it stabilizes a more active form and fine. All it means is making it more active, making it less active. So just a little bit to kind of wrap this up. Enzymes are oscillating. They do cycle between active and inactive forms, and you can sort of see it right here in this model simulation. You're seeing this thing sort of twist and turn. This has got a double subunit here, and there's something going on right there. That's kind of alluding to what we're talking about here. In any case, an activator will stabilize it in a more active form. An inactivator inhibitor will stabilize a more inactive form. And there are some other examples of this that are more advanced, like cooperativity, where you bind one substrate and makes the other sites more available, et cetera, et cetera. But again, activators stabilize, make the enzyme more active. Inhibitors tend to make the enzyme less active. But identifying potential allosteric regulators, whether they're a chemical molecule or a peptide or an antibody, is huge because these things demonstrate high specificity. One of the worst things that happens with drugs is that you may have a powerful drug, but it has side effects. The reason it has side effects is that it's affecting other areas of the body, other proteins that are similar to yours, and thus you have non-specific interactions going on. But if you can find an allosteric regulator that's specific, you have a very, very viable potential product. For instance, they talk about inhibiting proteolytic enzymes called caspases. These things are involved in cell inflammation and particularly cell death. You could find some way to manage caspases. Maybe you would get you could suppress inappropriate inflammation, and they're talking about that in the book. But on a more new level, this is a spike protein found on the surface of the coronavirus, the SARS-CoV-2 virus. If you could find an inhibitor for this, you can see that it moves. You could find some way to prevent this thing from interacting with a host cell. You could, in theory, cure people of a coronavirus. As it is right now, they're working on ways to develop antibodies against this spike protein, and it's a natural target because it's sticking way out there on the surface of the membrane capsule, which was formerly a phospholipid bilayer from the host cell that it came from. Those are phospholipids down in there. But again, very, very attractive. You could find a specific way to inhibit this protein because then it couldn't get into the next host cell. Back to the caspase example that I was just talking about, we know that these caspases have active sites and they can cycle between active and inactive forms. So we know that they have active forms from studies. And while we generally talk about apoptosis being non-inflammatory, the truth is that sometimes when it occurs inappropriately, it can cause inflammation and cell destruction. So if we can block those caspases, we can prevent the progression to cell death that we may want to avoid. So again, we have this active and we have this inactive. What we want to do is stabilize the inactive form. A substrate can do just that. We want to avoid that. What we do is we use this allosteric inhibitor to stabilize it so it never binds the substrate in the first place. And here they're showing the space filling models. This is the one with the inhibitor in there, stabilizing this inactive form. Another very common way of controlling chemical reactions. Again, it's allosteric regulation, but in this case what we have is the end product, or sometimes intermediates, coming back and shutting down earlier steps in the metabolic pathway. We call this negative feedback or feedback inhibition. Very, very, very common. The end product shuts down the pathway, usually at a relatively early step. It prevents the cell from wasting resources because you're not going to make D and E and F and G. You might need these for other things, but this is a committed, what we call a committed step. So once it goes from C to D, it's now going to go this way. Well, if we don't need this, we certainly don't need any of this. So G can come back and prevent the creation of letter D and thus shut that pathway down. It's allosteric regulation. We call it feedback or negative feedback inhibition. So here's just an example. We are manufacturing an amino acid called isoleucine. When isoleucine is created, isoleucine will feed back, block the enzyme, and prevent the creation of these intermediates. Okay. So it's an allosteric regulator. It's not a competitive inhibitor. It's an allosteric regulator, and if isoleucine is used up by the cell, it frees that enzyme to create more isoleucine. One more type of control mechanism that wasn't really illustrated in that earlier slide showing you the genetic and the allosteric, and that is localization. We can control whether or not an enzyme can do its function based on where we keep it in the cell at any given time, preventing loss of control or inappropriate reactions. And so that's what membranes do, and organelles do, and cytoskeleton does in order to help not only control how fast the enzyme works, but also control it in a three-dimensional way. Think of two boxers in a ring. You can control whether or not they're boxing either by forcing them into their corners, or if they're actively boxing, you can even have the referee get in between them and push them away. That's a kind of an analogous sort of a situation. I wouldn't want you to use that on an exam, but if you can see it that way, you can begin to understand what I mean by three-dimensional and spatial regulation of a reaction. So structures can do this, cytoskeleton can do this, and enzymes can actually serve both structural and functional roles in a membrane. For instance, respiration in the mitochondria. So let's look at that picture. And as you can see in this image with the mitochondria here and the electron microscope image, all kinds of folds and membranes. The main point of these folds and membranes are to create compartments so that we can regulate reactions. We can stabilize proteins in these membranes so that not only will the proteins be under control, but where their reactants and their products go can be under control. We can even see in this rendition, look at this membrane right here. This is an inner membrane versus the outer one, and you can see there's a different complement of proteins in each of these membranes in different compartments, each with molecules that serve different purposes. So control, 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 not only in terms of the shapes of the proteins, but where the protein is and how that protein is being managed are all aspects of regulating metabolism. And so that concludes Chapter 8. A lot going on in Chapter 8. The key is to be methodical. If something gets by you, take a little bit of time, go back, review it. I know that sometimes it can go a little bit on and on, but don't let yourself fall into that trap. You can counteract that with active learning strategies. So good luck, thanks for your time, and I will see you in the next chapter.